Is everyone able to see the screen okay?
Yes, you may want to hit present. So it's, yeah, there you go.
Okay, yeah. So thank you all for being here. It's a pleasure to present. And so basically, I'm going to be talking about how does experimentally induced plasticity affect functional connectivity in the brain? And this is the research question that I was addressing in the research process the past several months. And I took two broad concepts together, neuroplasticity and functional connectivity, and I related them in a very unique way, which I wanted to share with you. So just a little bit of background for the research process. What exactly are these two concepts? What is functional connectivity and neuroplasticity? So functional connectivity is talking about the correlation in activity patterns between various regions of the brain. Now, these can be homotopic regions, which are regions that correspond to the same network on both hemispheres, like a mirror, or non-homotopic, which are completely different regions on either hemisphere. So what is neuroplasticity now? Neuroplasticity refers to the altering, creating, or the destroying of neural connections within the brain. And these are most prevalent in the adolescent years, and these shape our habits and our behaviors on a daily basis. And these are two important concepts in the context of neuroscience as a whole, and I thought it'd be very significant to talk about these. So how exactly can these concepts be examined? There are a variety of neuroimaging techniques that can analyze activity patterns within the brain. And the technique that I looked at is fMRI, or functional magnetic resonance imaging. And this is basically tracking activity patterns across a variety of regions across the brain. And for this study, I looked at regions within the cerebral cortex. And it's basically so that you can take a slice of the brain and analyze activity across that entire slice over a varied period of time. So there are also a variety of techniques that can be used to analyze activity, such as positron emission tomography or transcranial magnetic stimulation. But I thought fMRI would be a great point of research for this process. So for my method, I basically used a quantitative research process, and I used statistical analysis. This included creating plots, subplots, color bar graphs, and more models. And I mainly did this in the integrated development environment, or the IDE called Jupyter Notebook. And the statistical modeling library that I imported in order to do this is called Matplotlib, which was very important for creating these models. And basically, I created these so that I can culminate into the final significance test for statistical analysis, which is a two-sample t-test. And what this does is it basically compares two groups, and it analyzes the means within these two groups to see if the change is significant or not. So one of the regions of the brain that I looked at, and it was actually based off of a prior study that was done last year, I believe, by Dylan Newbold and his team of researchers at Washington University at St. Louis. So he basically analyzed functional connectivity in the somatomotor system of the brain, and in particular, the hand motor region. And I basically was able to use a data set that took functional connectivity patterns, and I was able to use them to create statistical models or visual representations. And as you can see in the first three graphs, there are three participants from the study that I was able to grasp functional connectivity data from. And there were a varied number of fMRI sessions for each participant. These fMRI sessions were done in 30-minute intervals over a period of 2 weeks. And what the biggest takeaway from this is that you can see that there is a significant trend for all three. It starts off between functional connectivity of 0.5 and 0.1 or 1, and then that's before casting. But during casting, it actually drops down significantly near close to 0, actually. And in the fourth graph, you can see that I actually create some dividers for each participant between pre-casting, during casting, and post-casting. And for this study, I actually only looked at pre-casting and during casting. I didn't look at post-casting because I felt that that was more irrelevant, as it was more about how each individual's brain reacted and recovered to the casting process. And I wanted to look at the immediate changes as a result of casting in terms of functional connectivity. So this was a very important starting point for my research process. So what I also did was I was able to use a directory from the Human Connectome project that contained task maps, which actually laid out activity patterns across the cerebral cortex of the brain. And I also used a tool called the Human Connectome Workbench in order to visually represent this task map. And I was also able to collect these node pairs for various regions of the brain. The first node pair that I looked at was the visual cortex. And it's in the back of the brain in the occipital lobe and processes visual information. And the first three node pairs looked at homotopic regions of the brain, whereas the last pair was from non-homotopic regions, which are completely different on either side. The second node pair is the default mode network, which is actually most active when no complex task is being performed, or when our consciousness is the most subtle. The third pair is the somatomotor system. And what's actually important here is that this is not the dominant upper extremity region of the hand motor region, which was actually analyzed by Newbold. This is actually the lower extremity region of the brain. And the results from this are actually very important, as I will discuss. The fourth node pair is the dorsal attention network on the right hemisphere, as well as the salient or ventral attention network on the left hemisphere. Now, the purposes of these networks aren't clearly understood, but I thought that the fact that these were non-homotopic would be very important to perform a two-sample t-test on as well. So using Jupyter Notebook, I was also able to create plots and subplots for each participant in each node pair. And as you can see, there's a key for each pair, the first three being homotopic and the last one being non-homotopic. And in the research process, I was kind of startled because there weren't actually very significant differences in functional connectivity from before and during the casting paradigm was implemented. And what's also significant about this is that the fourth node pair, which is non-homotopic, is actually has the least functional connectivity, which makes sense because they're not part of the same network and therefore are going to have very different functional connectivity trends. And therefore exhibit different activity patterns. And from these visual representations, I was able to use a two-sample t-test for each participant and node pair combination for a total of 12 t-tests, which I've laid out for my results. And the black lines divide pre-casting, during casting, and post-casting groups, which will also be important. So these are examples of subplots for functional connectivity correlation between hemispheres for each node pair. So this is more specific than the plots because it shows exactly the intensity of the activity between each hemisphere. And the big takeaway here is that the first three hemispheres, or the first three node pairs, actually have very similar functional connectivity patterns, right? The orange and blue lines overlap pretty similarly. But of course, the fourth node pair, which is non-homotopic, has very different functional connectivity trends, which I thought was very interesting. So the culminating point of the research process was conducting two-sample t-tests as a result of these visual representations. So to go over, the hand motor node pair, which was the main node pair that was looked at from a Newbold study, which I visually represented and then put into a two-sample t-test. As you can see, all three of them have very significant trends. So this actually makes sense, of course, because the hand motor node pair was where the cast was directly placed on, and therefore it was disconnected from other regions of the brain. And therefore, there's going to be a big difference between before the casting and during the casting. The significant trends are represented in bold. So for node pair 1, which represents the visual cortex, the only significant change we saw was in participant 2 with a very low p-value. whereas the other ones had relatively high p-values, which I thought was interesting. So the null hypothesis for the t-test in general that I had was that functional connectivity differences from before and during the casting, any changes were actually due to random chance and not the locations of those nodes themselves. Whereas alternatively, any changes in functional connectivity from before and during were a result of something other than chance, or in this case, the actual locations. For node pairs 2 and 3, which include the default mode network and the somatomotor system, I saw no significant functional connectivity differences. And for node pair 3 specifically, this is important because it was within the same region as the hand motor node pair or the somatomotor system, but it still did not have any significant functional connectivity differences. And this shows that although two node pairs can be within the same system, they can still have very different functional roles and therefore exhibit very different patterns. Node pair 4, of course, was the non-homotopic region. And actually, the first two participants did exhibit two different functional connectivity trends, whereas the third one didn't. So you can kind of take away that for some participants, non-homotopic regions of the brain will exhibit significant changes if a certain region of the brain is plasticized. And for some participants, this may not be the case. So some limitations of this research process was the lack of node selection precision. Now, the Human Connectome project allowed me to select these node pairs by hand, so they're handpicked. But because each hemisphere of the brain has slightly different topographical features, there isn't a completely precise way to select node pairs so that the activity patterns will be exactly the same, right? So even a slight difference in location of selection can result in large differences in functional connectivity. The other limitation that I saw was a lack of participant data. So the fMRI dataset that I took actually, if I go back, contains only the 3 participants, but the larger study that this was based off of did have more participants, but their data was not included. So I think having more participants in this data can give a more firm basis for the results and therefore fully enhance the understanding of this research. Some implications of this research are that it's first of all just helpful for various neuroscience researchers who specifically look at how activity in the brain changes on a large scale. And since I looked at how neuroplasticity induces these changes, it's very helpful for individuals who actually have neurodegenerative disorders, some common ones being ASD, ADD, bipolar, or schizophrenia, because these individuals are confronted with lapses in brain judgment and activity. And so I wanted to look at how changing a specific region of the brain induces changes in other regions of the brain, which is very important. And because of this, it is a possible precursor for therapeutic interventions. So analyzing how exactly activity can change within these kinds of individuals can actually help for treatments for plasticizing other regions of the brain that can induce other activity patterns. And doing this can potentially fix these abnormalities in activity patterns so that they can function what we consider to be more normal. So yeah, this was basically what I went through for my research process. If you have any questions, let me know. And yeah, thank you so much.
Thank you, Arjun. I actually have a question just to follow up on my understanding of your project. You mentioned this casting. Would you be willing to tell me a little bit more about what you mean by the casting process so I can better understand those results?
Sure. So casting is a more broad term for actually functionally disconnecting or isolating a specific region of the brain. So it actually, it uses some magnetic stimulation to isolate that region of the brain. And so what it does is it completely blocks neural connections with other regions of the brain. Because all regions of the brain have some interconnectedness in terms of neural connections, and casting is actually just generally stopping those neural connections magnetically so that that region of the brain is on its own. And this induces changes in other regions of the brain as well.
Thank you. That was helpful. As you know, you have these formal oral defense questions that I'll get to. And so I will choose one from each of the three categories that you prepared for, which is the research process, your depth of understanding, and reflection on the inquiry process. So my first question for you is, how did your initial exploration of the scholarly conversation that you were looking into really kind of spark your interest into this research question? Could you tell me a little more about that?
Definitely. So when I first embarked on this research process, the one thing that I wanted to address within the context of neuroscience was actually neurodegenerative disorder. Now, when I read relevant literature, I was kind of wondering how exactly individuals with neurodegenerative disorders came to be, right? Is it a result of activity patterns, just abnormalities in structure? And as I kept reading, I came across neuroplasticity and functional connectivity, which actually did talk about their implications on neurodegenerative disorder. So what I was able to do was using the study by Newbold, which I had mentioned, I was able to connect these two concepts that really interested me into finding significant results and seeing how those results affected activity patterns and what that meant for individuals who might suffer from abnormal activity patterns. So really looking into these two concepts and following up on that through statistical analysis really furthered my understanding.
Thank you. So when you think, you've already addressed this to a certain degree, but I'd love to hear a little more about how your findings really do provide some direction for future research in the field and what comes next.
Definitely. So because of the limitations, actually, there could actually be more done to track functional connectivity trends. But as I mentioned, the functional connectivity patterns that I did see looked at, for example, the visual cortex, the default mode network, and the somatomotor region. So looking at these could actually be a really good precursor to further research into looking at not only individuals with neurodegenerative disorders, as I mentioned, but actually people with disorders that they have come about in their lifetime, such as ischemic stroke, or other regions that actually focused on, the motor region or the visual cortex. And if you look at these regions specifically, researchers can still analyze how functional connectivity varied within specifically those regions and not just neurodegenerative disorder in general. So coming up with treatments for enhancing activity within those specific regions can also help individuals with abnormalities within those specific regions and not just lapses in brain activity in general. So that's also how it could help.
Thank you. And my last question for you, if you could kind of go back and revisit your research process, what would you do differently and why?
Yeah, so I think one thing that I mentioned when I first started this research process was the actual technique of neuroimaging, which was functional magnetic resonance imaging. And I chose this because it was very versatile in terms of collecting brain activity. But what I thought that I could change a little bit better was using a different technique to analyze functional connectivity. And this was limited by what I used as a basis for my research, right? I based it off of a technique used by Newbold. And I thought that I could actually perform a technique of my own, such as positron emission tomography, as I mentioned. And I think this could actually deepen my results a little bit more and enhance my understanding, because fMRI is a very generalized technique, and many individuals use it not only to track activity, but just structure in general and track activity within other regions of, for example, the central nervous system, the peripheral nervous system, and other regions relating to the brain. So I thought that using a different technique could actually be something I could do differently.
Thank you. With that, it ends the formal portion of our presentation with oral defense. I'll stop the recording here, one moment.